Hello, everyone, and welcome to Symposium, and thank you very much for joining this presentation. Today, I'll be talking about the detection of CRISPR-related structural variants in genome-edited hematopoietic stem cells and X-SCID disease. All of this work has been done in collaboration with and led by Dr. Saksi Duraban from the National Institutes of Allergy and Infectious Disease. My name is Alex Hastie. I'm the Vice President of Clinical and Scientific Affairs at Bionano, and I'll be presenting this really on her behalf. Severe combined immunodeficiency disease, otherwise known as SCID, is a disease which has a phenotype of little or no resistance to infection. Patients with SCID are diagnosed based on immune cell count. They have very few or no immune cell, T cells or NK cells. And while they have B cells, they are not functional, the B cells. So as a result of this lack of immune cells and functional immune cells, patients generally succumb to infectious illness early in life. Some of the early treatments involved isolation, such as in a sterile bubble. And you can see a picture of David Vedder at Texas Children's Hospital, where he lived in this sterile bubble for most of his life. And for that reason, this disorder has also been termed bubble boy disease. One form of SCID, X-linked SCID, is known to be caused by mutations in a gene on the X chromosome, IL-2RG. There are a multitude of mutations which can impact this gene and results in improper function of the cytokine receptor, which it's a critical subunit of. And the cytokine receptor is critical to immune cell function. Therefore, those mutations will knock out the immune system. More modern treatments include bone marrow transplants. Initially, these bone marrow transplants were allogeneic, and those allogeneic donors, the donors for those transplants, needed to be matched siblings. These transplants needed to happen before the age of 3.5 months of age, and the recipient needed to be infection-free. Unfortunately, only about 10% of patients had a matched sibling donor, and even those that did have a donor had a low survival rate, mostly as a result of graft versus host disease. So to address this unsatisfactory outcome, gene therapy using autologous hematopoietic stem cells was developed. In this case, a functional ILGR2 gene is inserted into hematopoietic cells from the patient's own cells. So a biopsy, a bone marrow biopsy, would be taken from the patients, and a gene therapy where a functional IL-2RG gene would be inserted into those cells, and then those cells would be re-administered into the patient. And by doing this, the treatment avoided the graft versus host disease and the need for a matched sibling donor. There are multiple methods that are available and in R&D for administering this treatment, this gene therapy. Some of the different methods include retroviral and lentiviral vectors, and these insert a functional copy of IL-2RG into the genome. They cannot fix the endogenous gene, and they insert into the genome in semi-random positions. So there are safety issues, including activation of oncogenes and disruption of other important genes. More recently, CRISPR-Cas9 methods have been developed and are in clinical trials, are in R&D and in clinical trials. The first methods involve CRISPR-Cas targeted insertion through directed homologous recombination repair, where the CRISPR-Cas can identify the location in the genome where the insertion will go. More recent methods involve base editing, CRISPR base editing or prime editing, where the actual mutations in the gene that are pathogenic are actually fixed. And so the benefit here is that the endogenous gene is repaired. So the team at the National Institutes of Allergy and Infectious Disease has started with the CRISPR-Cas method. In performing any sort of ex vivo gene therapy, R&D and research, testing of the accuracy of the gene editing and any deleterious off-target effects is essential. So they tested for guide-dependent off-target effects using in silico predictions as well as several sequencing methods. And they also tested for whole genome guide-independent off-target effects using whole exome sequencing at high depth and whole genome sequencing. And finally, they tested for genomic stability using karyotyping. But because karyotyping is very low resolution, they also used optical genome mapping for a much higher resolution assessment of genome stability. In setting up this assay, we collaborated to first assess the sensitivity of optical genome mapping for detection of structural variants. We established lower limit of detection in our experimental design at 2 coverage levels. We mixed 2 commercially available cell lines that have known structural variants, such that those variants were at different variant allele fractions, starting at 10%, going down to 0.5%. And then we asked whether we could detect those variants at those different variant allele fractions. Using 1.5 terabases, or about 400x genome coverage, we could detect all of the variants at 10% and about 90% at 5%, and then the sensitivity starts trailing off. At 5 terabase coverage depth, or approximately 1600x genome coverage depth, we were able to detect all of the variants at 10 and 5% variant allele fraction, and 96% of the variants at 2.5%, and still 75% of the variants at 1%. So therefore, so now that we've established our lower limit of detection, we applied optical genome mapping to the samples that had been edited by the CRISPR treatment. So the experimental design here is that we collected high depth data for the high sensitivity assay from the CRISPR edited sample as well as the naive parental sample. And we compared the structural variants between the two to see if there were any variants that were induced by the genome editing. We saw no large-scale off-target structural variants in the genome. We did see some structural variants at the target site on X chromosome. Looking closer into this locus, the IL-2RG locus, we called an insertion at the target site. This is the expected insertion of the functional IL-2RG. We also reviewed the individual molecules, and we saw that there was likely heterogeneity in the insertion size. So we looked a little bit more closely at the insertion sizes. Here we've plotted the insertion sizes in the IL-2RG locus, and in the control we see no insertions, while in the genome edited samples, we saw that about 26% of the molecules had an insertion about 1 to 2 kilobase pairs in length. And then a few percent of molecules had longer insertions that could be concatemers of the gene editing cassette. We next looked at other structures at the IL-2RG locus. And here, unfortunately, we found an undesired structure that here we found an undesired structure. We found 2 reciprocal head-to-head and tail-to-tail fold-back fusions These structures appear to have very low variant allele fraction, approximately 1%, and are not present in the untreated sample. So what might this mean? There are some reports of similar structures in CRISPR editing. The fusion structure indicates that a pair of sister chromosomes, here showing the X chromosome, were cut at the target sites and then fused back together after swapping, translocating. So in that case, we have an X to XP to XP fusion, and another fusion that's dicentric, which has the XP to XP ends of the chromosome. So this results in a acentric and a dicentric chromosome, which is consistent with these fold-back fusions. Now, we next asked, are the cells that contain these abnormal chromosomes viable, and are the chromosomes themselves stable? So we grew out this culture for another 2 weeks to see if those fold-back fusions were still present. And we repeated the optical genome mapping in comparison to the untreated control. And in this experiment, we could no longer detect these fold-back fusions, indicating that the cells containing them are not viable and did not maintain in culture. We also looked at the insertion site to see if the insertion was still present, which it was. So although we were encouraged to see that the fold-back fusions disappeared and the insertion had been maintained, we also asked whether newer CRISPR methods like base editing would create these structures or would maintain better genome integrity. So we looked at base editing of IL-2RG. Base editing is a method that uses a CRISPR-Cas9 based nickase that directs a deaminase to a specific location in the genome and can alter the sequence at that location. And by doing this, we can precisely correct a single nucleotide mutation that's pathogenic and restore function. So the team at National Institutes of Allergy and Infectious Disease created this CRISPR treatment and treated hematopoietic stem cells to correct the mutation and showed that this restored functional IL-2RG. We analyzed this sample using optical genome mapping and again looked for off-target and on-target structural variants, and we found none. We looked right into the IL-2RG locus, and we found no abnormal structural variants, including insertions or fold-back fusions. So in comparison to the CRISPR-based homology-directed repair method, where an insertion of a transgene is used, this method seems to maintain stability of the genome much better. So in conclusion, optical genome mapping detects structural variants genome-wide at approximately 1% variant allele fraction using the high-depth workflow. For CRISPR-Cas directed gene insertion, optical genome mapping reveals on-target undesirable structural variants, these P and Q fold-back fusions that I talked about. That optical genome mapping detects insertions of different sizes and can provide relative quantitation of these insertions. And base editing for mutation repair avoids double-strand breaks and has no detectable induced structural variants according to our optical genome mapping analysis. So with that, I'd like to close by acknowledging Dr. Saxi Duravan, who's been a wonderful collaborator in this and really led this entire project. And then others involved in the project, such as Xiaolin Wu and the team at Leidos and Bionano. Thank you very much for your attention, and I hope you enjoy the rest of Symposium.